Thank you, Ollie, and thank you for inviting me. This is also one of my favorite conferences, especially since it's just down the street and the view is gorgeous. What I'm going to talk to you about today is a little, it's not quite as mainstream as what you've heard already, but it shows how you can combine two areas of expertise to do a better job at something that we believe is very important. So the topic I'm going to talk about is the development in my lab of a cell therapy for Parkinson's disease. It's a cell replacement therapy, and it is autologous, so it comes from the same people. The reason that it is important for us to have genomics is because of all the things that happen to stem cells that when you grow them, and also you'll see to identify them. Genomics is an enormously important tool for the development of this therapy. So I'll give you a short basis. I think most of you probably know somebody who has Parkinson's disease. I've talked to very few audiences that don't have some relative or some acquaintance. Parkinson's disease is caused by the loss of a particular type of neuron in the brain. It's a dopamine neuron. It lives in the substantia nigra, which is where that right here, this little tiny part of the brain, it's called the midbrain. And the cells in that area send their axons, their processes up into this area, which is called the striatum. And this is the circuit that is lost in Parkinson's disease. When these cells die, that innervation disappears. And that's what's responsible for the motor problems that are associated with Parkinson's disease. It wouldn't be surprising if it were also associated with other aspects, but that's all we know for sure. So the therapy right now is not great. There's a drug therapy called L-DOPA. Because it's a neurodegenerative disease, adding L-DOPA makes the remaining dopamine neurons, sort of hypes them up so they work much better. But the degeneration continues. And so eventually, since it's progressive, the rest of those neurons will be lost, and L-DOPA no longer works. For people who have some really serious physical problems, in maybe mid-stage Parkinson's disease, there's a technique called deep brain stimulation, in which electrodes are put into people's brains, essentially tuned so that the stimulus counteracts the tremor. That works great too for a while, but it's a progressive disease and the neurons will eventually die. So what we decided to do was not to try to figure out what causes the disease, but try to figure out the most simple way to fix it. And what we decided on about 6 years ago was that there was technology available to make pluripotent stem cells from individuals. They're called induced pluripotent stem cells. And my lab has started using that technology for a lot of projects, and we thought this was probably one of the best applications we could think of. In this idea is that we make these pluripotent stem cells, which can make every cell type in the body. You might not even want to leave the stem part in, because I know there are other kinds of stem cells. People get confused. These are pluripotent cells. And then from those cells, we generate dopamine neurons, which we plan to transplant back to the brains of the patients. So I'm going to do this really, because the same questions get asked a lot, I'm going to just propose them and then tell you how I've answered them. I think that will cut down a bit on the time later. And they're absolutely understandable, and I am looking for new questions, just in case you want to start thinking about it now. So why would we use cell replacement therapy for Parkinson's disease? The reason is that it's loss of a single cell type. So if you can replace a single cell type, then you should be able to treat the disease. And the reason why we chose dopamine neurons, not only because they get lost, but because there was a group of trials that were done in the 1980s through the 2000s that used fetal cells, the precursors of dopamine neurons from fetuses. And in some cases, the dopamine neurons developed and stayed in people's brains for decades. The reason we want to use autologous cells is not just because we can, but because we want to avoid immunosuppression. That means that we can put these cells into people's brains and they won't be rejected. People don't have to be immunosuppressed. And also importantly, if the dose is too small to start with, we can always give people more. Not that this is brain surgery, you don't want to do it every day. but in fact it can be supplemented over time. You can't do that if you have an immune reaction to the cells that you got. So here's the proof of concept. These are the fetal cell transplantations I told you about. Essentially, it involved taking a little tiny bit of a very young fetus. The fetuses were about 6 to 10 weeks old. And this little part right here in the midbrain contained the cells that would give rise to dopamine neurons, as well as a few other neural types. So what happened was these are put directly into the target of the substantia nigra. Now I have the nigra wiped out so that they don't have to grow anywhere to make synapses. So they're put straight into the target where the axons make synapses, and they make synapses in situ. So there were around 300 people altogether treated during this time frame. And they were all done independently, so it's a little hard to make the results all fit in. But occasionally there was long-term reduction of the Parkinson's disease in some patients. But there was a side effect, dyskinesia in others. And that was induced by the graft. It caused people to have uncontrollable tremors, uncontrollable movement. It looks like the reason for those dyskinesias was because of the lack of purity of the cells that were put in the brain. Because one type of neuron called a serotonin neuron does, in fact, cause that. Now, whether that was the cause in the fetal tissues, we're not absolutely certain in all cases. Now, this is one example in which this technology worked really well, fetal cell transplant. This was published fairly recently. And it shows that this is one side of the brain where they only got a transplant on one side, which is one of the problems. You can't get enough fetuses. The other side received transplants of several fetal tissues. And you can see the difference very easily. The brown stain here is tyrosine hydroxylase, which is a marker for dopamine neurons. And if you look up close, you can see that there are healthy dopamine neurons in the target. And they have to be graft cells, because there are no neuron cell bodies in that part of the brain that make dopamine. So we've learned a lot from that. In this particular case, the graft was healthy for 24 years. So that gives us optimism about if we can reconstruct this, it will be long-lasting and durable. And also the dopamine levels in the brain increased over time, which means these cells continue to develop. And we also got an idea of about around how many neurons needed to be transplanted in order to restore function. These are all valuable for our plans. So the bad things about this kind of therapy was that the good things were that the grafted neurons survived and that they restored dopamine signaling. The bad part was quality control. And really, it is all about quality control. And that resulted in inconsistent results from lab to lab, and in fact, within labs. And then, of course, there are growing legal and ethical issues now for the use of aborted fetuses. We can't do that in the US now. So instead of using fetal tissue, we decided to use pluripotent stem cells. This is my diagram of what pluripotent stem cells can do, this tree. These are the cells down here. They give rise to everything. In mice, they give rise to an entire mouse. Obviously, we haven't shown that in humans, but we have the same type of cells. So that means you can make anything. And there are lots of labs that are working on, working on, for example, heart tissue, and here are dopamine neurons and other types of brain tissue to make these cells and transplant them. These are the two sources. One of them is the embryonic stem cell route, which is taking a blastocyst stage embryo and isolating the pluripotent stem cells. And the other, which is the one we use, is to take a skin biopsy, grow up the dermal fibroblasts, reprogram those cells using Shinya Yamanaka's method, and end up at the same place. There are four, we're not alone, there are four groups, major groups in the world that are working on similar strategies using pluripotent stem cell-derived dopamine neurons for treatment of Parkinson's disease. And this is from a paper from 2017. These are the results of a meeting we had in Kyoto in which we shared all of our plans and our results. So this gives you sort of a snapshot of what's going on in the rest of the world. This one right here, Jun Takahashi's study in Kyoto, he has already transplanted one patient. He did so in December of last year. And he's using iPS cells, induced pluripotent stem cells, but they come from one person and they're not matched to the patient. Okay, so they're just like embryonic stem cells, they're not matched. And so they need to use immunosuppression. The group Loren Studer runs in New York, this is now Blue Rock, a company called Blue Rock. They also will use immunosuppression because they're using embryonic stem cells, which of course are not matched to anybody. And likewise, Roger Barker and Malin Parmar in Sweden and the UK are also using human embryonic stem cells. So I just want to point out we're the only ones who decided to go this route, and that it took us in a different direction than the other people. And this is where the genomics really comes in. Okay, so these are the questions that everybody asks. How do we know that we have the right neurons? Because these cells can make anything, right? So how do we know they're not just like, making something that doesn't actually exist in the body? And then we need to answer the question about whether these neurons are functional or not. And I'm going to show you the answers to this. We use a gene expression signature to identify the cells. I'll show you why we do that. And we link that to functional assays that we use in vitro, in which we look for the maturation of these cells, the release of dopamine, and their electrophysiology by using a patch clamp, so they have the right electrophysiological activity. We also look at their function in vivo, and this is done with to look at the behavioral recovery in a rat that's had half of its brain depleted of dopamine neurons. We are asking whether the human cells can compensate for that. Okay, so this has a lot of this is an overview of a lot of what we've done. You can see the stages. We start with fibroblasts, make iPS cells, differentiate them into immature dopamine neurons. And this is the stage at which we transplant the cells. This is important because if we let the cells go on to mature, they would not survive the transplantation or the cryopreservation because we freeze the cells as well. So if you look at these pictures in the upper right-hand corner, these are two different patient-derived cell lines. And these are stained with tyrosine hydroxylase. You can see that they have a fair number of positive cells in both. But importantly, just below that, you can see that when you look at the neurotransmitter production by these cells, both of these cell lines only make dopamine, and they don't make serotonin, which is that neuron type that we want to avoid. And then at the bottom, you can see the electrophysiology. We've done this for a number of different cell lines. Every single patient cell line has been successful so far. So this is how we designed our behavioral studies. We had a bit of trouble at first finding somebody who could do the rat behavioral studies. We finally found out the best people in the world were in Cardiff, in Wales. So we sent the cells to them, and we sent a person to them. They transplanted the cells to the brains of this particular model of rats. And this is the result. Essentially what happens, if you look at that picture, the picture here, one side of the brain, those are the human cells. This is the same stain that you saw before. These are the human cells. And this is the normal endings from the axons from the substantia nigra on the other side of the brain. So these are actually cell bodies, which you can see in this picture. Sorry, it's so pixelated. But what happens is if you give these animals amphetamine, they will rotate. They'll actually circle in the direction of the lesion because the dopamine is all released on one side of the brain, and it causes them to go in circles. So that's the behavior we're looking for the recovery of. In other words, we want to look to see when and if the human cells can balance out the input from both sides of the brain in the rat. And these data show that we can. You see that they continue to rotate if nothing happens. If they are controls with no lesion, they don't rotate at all. But these two are two different Parkinson's disease patients. And you can see that they start out with the lesion phenotype, and then over time, they come down to the control phenotype. So that means these cells have completely compensated for the rat cells. Okay, this is an important question that may seem a little bit obscure, but there have been people who reported that there were even though they're autologous cells, they may still be rejected. So we decided to ask that directly. Very quick slide. The way we did this was to test their antigenicity using a new assay that is not a standard immunology assay. It's a lymphocyte cell reaction. So we took the blood from one person, and then we challenged their T cells with the fibroblast iPS cells and neurons of either that patient or another patient. And the result would be if there's an immune reaction, then those T cells will proliferate and they'll secrete cytokines. So I'm just going to give you the final information about that. This is an overview of the results and whether we got this T cell proliferation or not. And you can see that whenever it's autologous, the same person's neurons are combined with their blood, they don't have any immune reaction. But if you see that if they're combined with somebody else's blood, they always have a reaction. So that shows two things. It shows that the autologous cells are likely not to be rejected, and it also confirms that neurons have the molecules that cause an immune reaction, which also was not clear to everybody. Okay, are they safe? Now we're getting into what the FDA wants to know. Are they safe? We've done animal studies to show that the cells don't form tumors. We can't detect any proliferating cells in those transplants. They're post-mitotic. And we also, with genomics, do tumor-associated genes by RNA-seq and whole genome sequence. We look to see that the cells are genomically stable using whole genome sequencing and Snip genotyping. And then we're using a gene expression assay to identify the cells at several stages. And I'll show you briefly what that entails. So this is an overview of the predictive genomic tests that we're using. Whole genome, and you can see at each stage that we use from the somatic cells, we sequence the fibroblasts and do Snip genotyping. And then we sequence them again after they've made pluripotent stem cells, and then once more before they are transplanted. We also do Snip genotyping to look for duplications and deletions. It's actually complementary to the whole genome sequencing. But I think importantly, we identify the cells themselves by gene expression profiling. This was the first effort of ours to define cells, predict what a cell would do based on its gene expression profile. This is called Fleury Test. It was published originally in 2011. It's very popular because we put it on the web and we made it free. So it's an application that allows you to just upload your gene expression data, usually RNA-seq data now, and then you get a graph. You get a list of information in a graph like the one that's on the right. And then you can just plunk that into your publication and you're done. You've shown your cells are pluripotent. So you can imagine that this has been very popular because the alternative was to inject the cells into a mouse and wait 4 months and see if they got a tumor or not. Okay? So this defines and predicts the fate of cells. They are pluripotent. That means they will form every cell type in the body. So this is our similar assay, which we call Neuro Test, which is the prediction of dopamine neurons to mature and function in vivo. It's very similar. We use a similar algorithm, but it's based on a reference data set. For Fleury Test, we had about 400 lines that had been confirmed to be pluripotent. And we used that to develop the algorithm that asks whether those cells match that type of cell or not. So we're using a neurotransmitter release. This is an HPLC assay of mature neurons to prove function. And we're assaying the cells at the transplant stage so that we can use this as a release criterion for our transplantation with the FDA. So genomics is really important because deleterious mutations occur in pluripotent stem cells. You may have missed the headlines, but there was maybe 20% of the embryonic stem cell populations in the world carry p53 mutations, which we all, I think, know are carcinogenic. So that's not a good thing. The cells make a lot of genes. They express a lot of genes, so we can screen them for cancer-associated genes. We can look for the mutations. We have a pipeline for looking at all the cancer-associated genes that we think will be important. And we just get that out of our Fleury Test data. We don't have to do anything else. We also use RNA-seq data from Neuro Test to identify mutations. That also allows us to look for contamination. You're just counting the transcripts. You can tell whether the transcripts came from neurons or from cells that were undifferentiated and therefore potentially tumorigenic. Okay, so our whole genome sequencing for deleterious mutations is based on a paper we published a couple of years ago. And essentially, it's pretty straightforward. We compare the cells with the donor fibroblast and look for changes. We also look for the appearance of any developmental or cancer-associated genes. So far, we haven't found any. Genetic variation in dividing cells is normal, so we expect to see them change over time, but the changes so far have all been benign. The Snip Genotyping is based on a paper we published quite a long time ago in which we used Snip Genotyping to show that there were common duplications and deletions that occurred as you grew pluripotent stem cells. And I'm not going to go into this a lot of depth, but if you look at all the red lines there, those are all reported in cell lines, duplications of parts or whole chromosomes. And there's one in particular that's very interesting on chromosome 20. The next slide, this is an example of the results of Snip Genotyping. We have we have on the top, we have the early passage of an embryonic stem cell line. You can see there's a straight line across. If you look in the same place, when the cells have been cultured for a long period of time, you can see it separates into two lines. That's because you have an extra copy of one of those genes. Okay? Here's my last question. What do you suppose the FDA thinks of this approach? Now, I have not, this is all in innocence. I have not yet done my pre-IND meeting with the FDA, but I have at every opportunity talked with people from the FDA. And the last time I did this was in September, in which they invited me to give a science talk at their working group on cell therapy. So the regulators were there. And this is an email I got back. It said, you represent an exceptional example of doing things the right way, and we want to help you along that path. So that's the best news I can possibly imagine getting from the FDA. These are the people in my lab right now, collaborators and the people who support us, including the ones who got us started on this project, a foundation called Summit for Stem Cell Foundation. My new company is called Aspen Neuroscience. We have existed since November, and we're carrying on with this project with investments. We do a lot of other things. Besides Parkinson's disease, we study multiple sclerosis, Fragile X syndrome, and we're also working with the San Diego Wild Animal Park to try to rescue the northern white rhino. So this is a picture of my lab on one of our outings to see some of our collaborators. Thank you.